All right. So, protein synthesis and gene mutation. So we have talked already about transcription and translation, but now we're actually going to talk about more in depth how the genes code for amino acids. Okay? And we're actually going to be able to take a piece of DNA and code it into amino acids and proteins. Okay? All right. I'm not going to read that. So, protein synthesis. DNA provides the instruction for how to build proteins, right? It's the master blueprint for how we are made. Master blueprint, then it gets turned into the messenger RNA, which is your working blueprint, so on and so on. Each gene dictates how to build a single protein in prokaryotes. It's open there. The sequence of nucleotides, the A, C, T, and G, of DNA dictates the order of amino acids that make up a protein. Okay, so remember, it's red. They decide which proteins to put together, and you end up with a complete protein at the end. Okay? All right, so protein synthesis, remember, occurs in two primary steps. Transcription translates the RNA or the DNA into RNA, and then translation takes that RNA and turns it into proteins. So the transcription creates messenger RNA, which is a copy of the gene to be synthesized. It occurs in, guys, I'm teaching right now. Corey, go to class. Okay, so transcription occurs in the cytoplasm of prokaryotes and the nucleus of eukaryotes. Okay, so it would occur in the nucleus of our cells. Translation is the messenger RNA being used by the ribosomes to build proteins. The ribosomes attach to the messenger RNA and use its sequence of nucleotides to determine the order of amino acids in the protein. So we'll look more in depth at how this works and see how things are coded. This happens in the cytoplasm of prokaryotes and eukaryotes. So after transcription, the mRNA moves into the cytoplasm and translates into the protein. Okay, so transcription. This is just going to go a little bit more in depth than what we've looked at. So it starts with the initiation. The RNA polymerase binds to a region of DNA known as the promoter, which signals the start of a gene. Okay, so it doesn't just grab onto that DNA willy-nilly. It finds that promoter and knows where it needs to start the DNA, or start the transcription so it can make the mRNA, so it can make the protein. The promoters are specific to each gene, and the RNA polymerase does not need a primer, so it doesn't need an area before it can start creating the mRNA, before it can start creating the RNA. Okay? I think it's just going to be the same slide. Okay? So the transcription factors assemble and the promoters at the promoter, forming a transcription initiation complex. The activator proteins help stabilize the complex. And gene expression can be regulated, turned on or off, by controlling the amount of each transcription factor. So the transcription factor tells that RNA polymerase how much it needs to create, how many proteins it needs to create. Okay, so Transcription elongation is the next step. So first we initiated, next we're elongating, which means it's going to start unwinding that DNA by breaking the hydrogen bonds that are between your bases. Okay, and the two strands separate from one another. Base pairing occurs between incoming RNA nucleotides. So remember, what are RNA nucleotides? Do you remember? DNA nucleotides are A, T, C, and G. What are RNA nucleotides? G. Okay, so the only one that changes between the DNA and the RNA are the T and the U. So there's no T's in RNA, there's no U's in DNA. Okay, so when you're coding, if it comes across an A, which would normally bind with a T, it'll put a U in. If it comes across a T, it'll put an A in. Okay, so again, remember it says, recall RNA uses uracil, which is the U, instead of thymine, which is the T. Okay, so it's using that template of the DNA to create the RNA that will then move on to translation. Good. Okay, so the U matches that, or the A matched up with U, G and the C, the T and the A, and then the C and the G, the A and the U, the T and the A. All right. Okay, and then just this bottom guy, the RNA polymerase catalyzes bonds to form between the ribose 3' nucleotide of mRNA and the phosphate of the incoming RNA nucleotide. Okay, so it's starting those bonds. So the polymerase keeps going through. Okay, and we'll eventually end up in the entire RNA. Okay, Whitney, make sure you're paying attention. I don't want to have to give you these notes. Okay, the gene occurs only on only one of the DNA strands. Okay, each strand poses a separate set of DNA, or a separate set of genes. Okay, so if it goes down one side of the strand, okay, it's going to make certain proteins. When it goes down the other side of the strand, it makes other proteins. Okay. All right, and then finally termination. So termination is just what it sounds like. It finishes. So a region known as the DNA terminator sequence signals the stop of the gene. Okay, so the RNA polymerase comes across that terminator and it leaves the DNA. It leaves the DNA and the mRNA, and then you're just left with your strand of mRNA floating around. Okay, so sometimes there can be some splicing. So exons are coding regions, introns are removed. Different combinations of exons form different mRNAs, resulting in multiple proteins from the same gene. Okay, humans have 30,000 genes, but are capable of producing 100,000 proteins. So that just means that depending on what's being created, different parts of the DNA will be cut out and spliced together to make different genes. Okay, and I believe that's usually done if there's junk DNA, because there's a lot of DNA in your body, not all of it is used. And the stuff that's not used is called junk DNA. So the junk DNA is cut out and you splice together what you need for the good DNA. All right, and then translation. So that's the second step in creating a protein. So we transcribed to the mRNA. Now the mRNA is translating to the protein. mRNA is used by ribosomes to build proteins. Okay, so it leaves the nucleus, goes into the cytoplasm, finds some ribosomes, and begins to create the protein. In the cytoplasm, some proteins feed directly into the ER of eukaryotes. So that just means that it's going into that rough endoplasmic reticulum and creating other things. All right, so this is where things become important, where you need to watch how things are being done, because this is kind of what your assignment's going to be on. So every 3 mRNA nucleotides are called a codon, okay? And each 3 specify an amino acid. Okay, so it starts with the promoter gene, and then every set of 3 nucleotides after that codes for a specific protein. Okay, so this CUA on the mRNA codes down to make the lutein. Okay, the next 3 will code down to make the next protein, or the next amino acid that goes onto that protein. This looks crazy. It's really not that bad, I promise. tRNA have anticodon regions that specifically bind to its codon. Okay, so the transfer RNA grabs from the mRNA, finds which amino acid it needs, and binds them together. Remember? So this is GAU. So we start G, second position is A, last position is U. Okay, so it's right there. And there's a different chart that we're going to use to figure these out, and I will show you that soon. Okay, so tRNA carries this specific amino acid. So the tRNA binds with that mRNA, carrying the amino acid, which then groups them all together and makes your protein. Okay, so the start codon is always this. AUG, that's always your start codon. Okay, so if I give you a big long list of nucleotides together, you don't have to start until the mRNA codon is AUG. Okay? So the ribosome moves from the 5'. Remember we talked about in DNA, mRNA is 5', 3', depending on which way you're moving down. Reading the mRNA and assembling amino acids into the correct protein. Okay, so it goes down, it reads them, it finds the codon, the tRNA that carries that amino acid, matches, and they bind together with those peptide bonds. Okay, and you see it continues on. The tRNA is bringing different proteins. Those proteins are joining together to make a polypeptide chain. Okay, so the ribosomes disengage from the mRNA when it finds the stop codon. Okay, so if you notice on this table, there's 3 places it says stop. Okay, on the table that we're going to use, and it is in this, it also says the stop. So there's 3 different codons that will cause this disengagement and it to stop creating the protein. We will do some examples, I promise. Okay, this is the table that we're going to use. Okay, so translate the following mRNA sequence. Okay, so first we want to break these into 3s. Okay? So, we have mRNA. mRNA will transfer right to your amino acids, to your proteins. Okay? So, there is no start protein, because that should be AUG, but we're just going to start anyways. So, A, G, C. So, A is my first, G my second, and then C, right there, is my third. So I have serine. Okay? And serine is abbreviated SER.
Does it have to be like that little box, or include the lens there?
What do you mean?
There's one right there that's like A, G, and there's a C in there, up there. But
That's a G.
no, I know, but never mind.
Here, let me get you a big copy of this so you can see.
I meant the one up there. Glycine.
Okay, which one are you talking about? Go point.
So, for this A, it just has to be the ones in here, right?
Yeah. So,
They can't like choose them with like the G and the
no, if you're, so this first letter is A. Okay, so it has to be in that pie witch.
A. Yeah, that's what I meant.
Okay, so you start with at the A, you go to the G, and then you go to the last letter, which is C, and you have your amino acid. Okay, the next one is U, so we're going to start at U, then A, then is that a C? Yeah, and then a C is there. Okay, so thyrazine. Okay, and then thyrazine, okay, is TYR. Okay, so okay, so I've given you all a codon. I want you to figure out what amino acid it is. Okay, so I've given you a codon. Figure out which amino acid it is. You'll need your page. Which one did you say?
I put on serine or whatever you said.
For A, G, U. Yeah, serine. Good. Okay, Jade, have you figured yours out? What is it? Serine. Okay, Dina?
Histidine.
Histidine. Okay. Okay. Good. Yep. And Whitney, have you figured yours out? Proline. Okay, CCG. Good. Yeah, everyone got them. Okay, kind of makes sense? It's pretty simple. Okay. All right, so this is just going to go through. So we had the serine, right? Okay, then the tyrosine, histidine, threonine, histidine, proline, serine, serine, serine. Okay, so there's more than one way that you can code for a specific protein. And if you look at the table at the bottom of your codon wheel, okay, it gives you every single way that you can code for that specific amino acid. Okay, there's many different ways. Okay. Okay, so serine. So that's just going through those letters. I don't need you to write this line out. I just need you to write that line out. Okay? Okay, and then they put those letters, the single letter codes, just to have even fewer things to write down. All right, so protein synthesis. Okay, so multiple RNA polymerase can engage a gene at one time. Okay, so it can be going through more than once, creating more proteins. Multiple ribosomes can engage a single mRNA at one time. So that's just making those proteins faster and faster and faster. In eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. Okay? In prokaryotes, transcription and translation occur simultaneously in the cytoplasm. Okay, and then this just talks about the different types of RNA. I don't know how much you have to worry about those. Okay? Cool. No video today.